This is the first part for chapter 22 on carbohydrate metabolism. And we're going to look briefly at what is digestion. So, first stage in catabolism is digestion, breaking down the food into small molecules. So it goes from physical grinding, softening, mixing of the food, but also the action of enzymes. So you have hydrolysis to break down those molecules of food into carbohydrate, proteins, and fats. Then things will continue, and eventually the product of digestion are small molecules that can be absorbed from the intestinal tract. This is made mostly by villi. We have millions of villi that are going to exist in the intestinal tract. And what they do, those villi are going to increase the surface area. That can be as big as a football field for humans. What happened then? Well, once they get into the bloodstream, the small molecule can actually be carried into the cells who need them. So this is a summary, and I really invite you to review it very well, understand it well, before we go to what happened for specifically monosaccharide in the bloodstream. So we're going to look at the glucose metabolism. That's here the overview. This is the major fuel that we need for your body. Fuel for the brain, working muscle cells, and red blood cells. What happens? Well, when the glucose goes inside a cell, so when it enters a cell, it is converted to a very exergonic, so releasing energy, glucose 6-phosphate. Interesting here, that's going to actually help trap the glucose with the cell because phosphorylated molecule can't cross the cell membrane. So once that glucose is trapped within the cell, it has different pathways. Now you're going to review those. That's the overview. So I really insist learn that diagram because it explains things very well. So you have the glucose on the top. What happened? Well, it really depends if your cell needs energy or if your cell has energy. So we're going to look at here a different step where it can be converted to glucose 6-phosphate and follow pyruvate, acetyl coenzyme A, then enter the citric acid cycle. This is when the cell needs energy, or is going to be stored into glycogen, and that's what we call glycogenesis, the making of the storage of glucose. Now, when we need energy and we don't have any more glucose in the bloodstream, we are going to tap on that stored glycogen. So that's going to be glycogenolysis. You break down the glycogen to get back into the glucose 6-phosphate and make the energy. You also have here the pentose phosphate pathway that can also exist and will be preferential to mostly do 5 carbon sugars that are going to be very helpful for nucleic acids. And we're also going to talk about what happened in anaerobic condition, what you have right here. Anaerobic means there is no oxygen, and you will have here building up of lactate. So review also this table because you're going to need them to really understand the function, and you need to know both the name function and some of the derivation of the name. And just here some overview. Well, as I say, when energy is needed, the glucose 6-phosphate goes through glycolysis, breaking it down into 2 pyruvate. Each of them will become 2-acetyl coenzyme A. Each of them will go through 1 round of citric acid cycle. But when the cells do have a lot of glucose, well, the excess, as I said earlier, is converted to glycogen. That's what we call here glycogenesis. Glucose 6-phosphate can also enter the pentose phosphate pathway, which is necessary for synthesis of nucleic acids.